Welcome. Good morning, good afternoon. On behalf of PHUSE, I welcome you to the Wednesday webinar. Here's a brief agenda that we are going to follow today. Happy New Year, first of all. I'm going to give an update from the Board of Directors. I'm Sangeeta Bhattacharya, the PHUSE Membership Director. You will hear from Mouly about the PHUSE US Connect 2020, which is just upon us. And then we'll hear from Delphine and Hiren on lessons learned from an FDA real-time oncology review pilot program. And we will end the webinar with Patricia talking to us about the technical conformance guide update on the use of simplified ts.xpt file for non-clinical studies. So US Connect 2020, we are almost there, 6 weeks to go. There's still time to register for the event. Our hotel has been sold out, but PHUSE is working to secure another blog booking. So details will be published on the PHUSE website pretty soon. Just the reminder of the date, it's from March 8th to 11th in Orlando at the SeaWorld, Florida. So look forward to seeing you all there.
Hello, Sangeeta.
Yes.
It's Mouly.
Yes, Mouly.
Are you intending to share any slide or slide deck?
Yes, I'm sharing. Lauren, can you confirm?
Yes, you're sharing, yeah.
Okay.
Ooh. So am I the only one who is not able to see then, I guess? Oh, yeah, that's okay. Sorry to interrupt, Sangeeta.
No problem. So then I wanted to also talk to you about the Computational Science Symposium, which is scheduled from June 7th to 9th at the Silver Spring Civic Center in Silver Spring, Maryland. Please save the date. As you know, it's going to be a really active and engaging meeting as every year. The sponsorship opportunities are still available, and if you are interested, please contact events@phuse.eu. The registration and call for posters will open soon, so I'm hoping that everybody is thinking about their presentations. Next is EU Connect 2020. Feels like we were just in Amsterdam in 2019. Our 2020 EU Connect will be in Liverpool. Call for papers will open in March. There will be sponsorship opportunities. It will be from 8th to 11th November, and we look forward to seeing you there. We have some good news and updates from the working groups. Anyone who has written a white paper knows it may not be extremely easy to put your thoughts together when you're writing a white paper. So the working groups have come up with, the PHUSE steering committee has come up with guidelines on how to write a white paper. Suggestions from length of the paper and writing style to how best to keep your audience engaged are all useful tips you will find there. You'll find a wealth of information to help you and your project on the working group's website, templates for presentations, our welcome pack, which provides a full overview of all our project, a who's who, together with training guides and helpful links are available. We have a request from the non-clinical topics working group. They would be really grateful if we could take a moment to assist in the annual data gathering by completing a short survey on industry standard for exchange of non-clinical data readiness and implementation. Of course, you'll find us on Twitter, we have on LinkedIn. Please use your hashtag PHUSE when you are posting. And thank you all for your engagement. And with that, I hand it over to Mouly to talk about US Connect 2020.
Thank you, Sangeeta.
Mouly, I'm sharing your slides.
Okay, thank you. I'm still waiting. Yeah.
If you go ahead, Mouly, I'll move on to slide 1.
Okay, all right. Good morning, good afternoon, and good evening for some of you if you have joined from that part of the world. Thank you everyone for joining the webinar. So PHUSE 2020 US Connect, 7 weeks to go. As you can see on the slide, it's from March 8th to 11th, and you cannot beat the location. It is sunny Florida. We're talking about Renaissance in Orlando at the SeaWorld. So yeah, as I said, cannot beat the location, and especially in the month of March. Now, some of the highlights for the conference are we have selected the best possible 14 streams that are relevant to the users. And in fact, of those 14 streams, we have about 164 presentations. Lauren, can you go to the next slide, please? Yeah. 164 presentations and about 30 posters. And Lauren, can you hear me?
Yes, we can hear you.
Are you on slide number 2?
Yes.
Okay. All right. Yeah, just want to make sure. So one of the highlights that we have are introducing to the US Connect is the PHUSE app. So it is going live. In fact, it went live, I should say. Sorry about that, on 13th. So one of the benefits of the PHUSE app is that you will be able to view the agenda. In fact, instead of having, let's say, a paper in hand to go through the schedule, instead of that, you could just probably view the agenda on the app, and then you could set reminder notifications for any of the sessions that you are interested in. So, and in addition to that, let's say if you have any information that you want to pass on to other attendees, conference attendees, saying that, okay, there is a session coming up, or maybe you attended a session which is interesting, if they want to join, you could do so through the PHUSE app. And as you know, during a conference, we always have more information, live information that we want to keep up with, and you could get that information through the PHUSE app, receive that information live about the conference through the app. One other key thing, which is kind of easier to work through the app, is exhibitor and sponsor profiles. Let's say if you have just limited time to visit just few exhibitors, you could just shortlist the ones that interest you the most by looking at the profiles on the app and go to those exhibitors. Then about registrations, we have already reached 500 plus registrations. It's not like we will be able to accommodate unlimited number of registrations. There is a cap. So what I would request and recommend is in your networks, whether it is LinkedIn, we are talking about organizations forum, or any of the vendors with whom you work, let them know that to register as soon as possible, because I'm pretty sure that since US Connect have been pretty popular and it's a great destination, location is great. Orlando, we are talking about, it will be sold out. The sooner they register, the better it is. Lauren, can you move on to the next slide, please?
Yep.
All right. Then talk about other highlights. We had introduced the summits, Executive Pharma Summit and CRO/Provider Summits in past US and EU Connects. In fact, I was honored to represent Seniors Health, the company where I work, as for the CRO/Provider Summit, and it's an excellent conference discussion. So all industry leaders, they kind of gather and discuss the most relevant topics and tackle the specific topics and see how that information could be disseminated to others in the industry. Now, one other event which I would like to highlight, and it's really important, is the PHUSE/FDA Data Science Innovation Challenge. So this is scheduled for all day on Monday, and the key is like we are trying to, the reason for this is we are trying to explore on the new technologies, kind of solicit information from the programmers, or if you will, or any other technical or functional users who attend the conference, and come up with some ideas and see if any of those ideas, if we could apply to the FHUSE working group projects. Kind of like the whole entire theme is to promote innovative thinking. Then moving on to the next slide is the keynote speakers. We do have a couple of keynote speakers. First one, Michael Rogers, so best-selling author, technology pioneer, and futurist. And the second is Ricky Kalman, mindset expert, motivational speaker, and celebrity hypnotist. So keynotes, they are really important. We have both keynotes, one on Monday morning, March 9th, and the second one is Tuesday morning, March 10th. I realized that at conferences, we tend to end up partying the previous night, but I highly recommend that, please do attend the keynotes. Don't miss out on the opportunity to hear from these excellent speakers in live. Then talking about the events, evening events on the next slide. So on Sunday, March 8th, we're going to have the kickoff with a poster session and drinks reception, which will be followed by buffet dinner. And all of this is sponsored by Indigo Group. And on Monday evening, the 9th of March, we have the FHUSE Connect dinner, sponsored by Summit Analytical. On Tuesday, the 10th, like previous years, again, we have scheduled to have the poker night. In addition to various skills, each of the attendees could bring, I mean, it could be technical, functional. I'm sure a lot of us have even these great poker skills. It could be Texas Hold'em or whatever, but I would recommend that please do attend. It's a location where you could lighten up, mingle, and have fun with other attendees at the conference. All right, then moving on to, I think this is my last slide, sponsors and exhibitors. So as we know, without sponsors and exhibitors, an event like this at this magnitude, it is difficult to organize. So we do have sponsors. Platinum sponsors are Accenture, Navitas Data Sciences, and Pinnacle 21. There are other sponsor categories, which I have just listed. There's no way I could have listed all the sponsors in various categories. So just to name a few of the categories, one is Connect, like the conference, US Connect sponsors, then app sponsor, evening event. I already went through the sponsors for the evening events in the previous slide, so that's covered, and then the catering. Now, an important element to a conference is exhibitors. And as you see, we have about 40 plus exhibitors signed up. So we will have booths where you could go and visit, solicit information on anything that you are interested about their organization, company. So I would recommend within, as I said earlier, reach out to your network, let them know that there are 40 plus exhibitors who will be present at the conference. So it's going to be a wonderful conference. So once again, thank you so much for your time. 7 weeks to go. Looking forward to seeing most of you at the conference. I think I'm passing it on to Delphine and Hiren. Lauren.
Yep, thank you.
Thank you.
Hiya, can you hear me and see my screen?
Yes, we can.
Okay, great. So I'll get started as there's quite a lot to cover. So hi, welcome to our presentation. And as mentioned at the beginning, this is covering our experiences on our tour from Novartis and Roche. So the agenda for today will briefly, very high level, cover what our tour is, provide our experiences, challenges, opportunities, and also end with some recommendations. Okay, so RTOR stands for Real-Time Oncology Review. So this is a pilot program launched by the FDA. And as the name suggests at the beginning, real time, the idea is to have this early engagement between FDA and the sponsor to see the analysis and the data earlier than the actual filing date. So the idea is, although not always guaranteed, but the idea is that this could hopefully lead to faster approvals and the treatments going out to patients sooner. The key thing we did want to highlight was there is an eligibility criteria. So here, we can't have every single filing going to apply for RTOR. And the idea of this is that, they are looking for straightforward study designs, endpoints that can be interpreted easily, substantial improvements over therapy, et cetera, et cetera. So they are looking for straightforward study designs. And originally the recommendation was for sNDAs, but I know there is an update to include NDAs as well, where it meets criteria. Finally, like I said, this is a pilot, so this will evolve over time as well. And over to Delphine.
Hello, everyone. So on this slide, I'm going to present the RTOR pilot timeframe as it is available on FDA website at the moment. So I will not go into each and every details because there are lots of things to cover, but would like to highlight a few points. So first, the starting point, which as you can see is the top line data availability or the database lock, or when the sponsor requests for RTOR participation. So when the sponsors contact FDA to request for RTOR, within 2 weeks they will get the answer from the Oncology Center of Excellence. And if it's positive, the first package will be sent out about as early as 6 weeks after the request came to the FDA. So just to highlight 2 points dataset-wise regarding the package. So you will see that expectation is to have all SDTM datasets sent, as well as key ADAM dataset for efficacy. But as you will hear during this presentation from Hiren's and my experience, things went a little differently. So I'm going to now present my experience in Kisqali. So yes, so I tried to summarize months of work in one slide. So it's quite something. So Kisqali was driven by Kisqali sNDA submission was driven by 2 pivotal studies, so Mona Lisa 3 and Mona Lisa 7. In this slide, they will be referred as M3 and M7. So by January 2018, so about 2 years ago, both studies met the primary endpoint, and results were shared with the FDA. So team was working on the submission and the filing when early April, FDA contacted Novartis, introduced us with the RTOR pilot, and Novartis agrees to participate. So following this meeting, we had lots of internal discussion. We also had few interaction with the FDA. So from datasets perspective, for both studies, the dataset were studies compliant and final. But documentation-wise, we were a bit behind schedule. They were not ready. So we were trying to figure out what was the best way to move on with the RTOR without the appropriate documentation. So we had few interaction with the FDA and decided to go with an alternative approach. So end of April 2018, the first package was sent. So it includes ML3 and ML7 SDTM, so all SDTM for both pivotal studies, as well as key efficacy and safety ADAM datasets. So we sent some datasets, but not the expected documentation because they were not ready at the time. SDTM were very standard and very similar to the original NDA, so we haven't sent any documentation. And for the ADAM, we created ad hoc specification, meaning that we describe in an Excel file the key variables and the main derivation rule. Afterwards, there was additional batches sent from other function. We also, at programming level, we had to deal with some information requests while we were finalizing the CRT package and work on all the submission deliverables. On June 28th, the full dossier was sent to the FDA. We were also working on the safety updates, so the data sets were shared with the FDA early July 2018. And on July 18th, we were very happy to learn that Kisqali SNDA has been approved. So it was very, very nice day for all of us because everybody worked tirelessly to get things done, to address questions. So the outcome was great and very quick because, as you can see, it was less than 3 weeks after the filing. So it's surprising as well for all the team members. So just a key message from this slide is, yes, the Kisqali has been approved less than 3 weeks after the filing, but FDA was able to start reviewing the key data of the pivotal studies 2 months and a half prior to the approval. So now I'm going to let Hiren introduce his experience.
Okay, sure thing. So thanks, Delphine. So for Venclex, the story was a little bit different, and I'll try and share it. I've got timelines, but I'll share it another way. So a lot of our work actually happened prior to the top-line results, and the reason was when the announcement came out, we were approximately 4 to 5 months or so prior to top-line results. So the questions were being asked to us, and they were rightly so, because a lot of the eligibility criteria boxes, our pivotal study was ticking them. So the questions were being asked, potentially, could we use our pivotal study, our filing as part of our tour? But of course, this was being done at risk. So a lot of the pressure on top of the filing work, the question was put to us, let's do this on top. And, we still may get a no at the end of this. So remember, as Delphine said, within 1 or 2 weeks after submitting a request for our tour, they could say no. So that was the biggest question that we were pondering, which is, should we go ahead with this or not? And ultimately we did. One of the biggest programming challenges also for us was trying to understand the language that was used, and the only real guidance, some of which we shared earlier, was referencing CDIS terminology. Now, the challenge with that was our study was non-CDIS, always planned to be non-CDIS as it was pre-mandate. So, for us, we preferred to provide it as non-CDIS. Many of the filing team members felt that, we should go down the full CDIS route. And when we did the feasibility, the challenge would be that it would take us beyond the original filing timelines if we converted everything to CDIS. So we were in a bit of a position where we needed to find another solution that could fit everyone. And that solution ultimately was something we'd find out from another project filing team, which was basically a term or a terminology that we coined as CDIS acceptable. And the idea was that we would convert our SDTM-like datasets to SDTMs. We would provide an ADSL to avoid the technical rejection criteria, but the rest of our analysis would be non-CDIS. So ADAM-like, if you want to call it that, but it would be non-CDIS. So using this approach, if you want to call it a compromise, we felt that we could meet the timelines with this approach, and it would still be really hard, but we would be a lot more comfortable than converting everything to CDIS. So that's what we went ahead with. So I'm now going to fast forward all the way, we've gone through our programming analysis, our specifications, our pooling, supportive study, conformance checking, etc., all the way to top-line results. We unblind, we meet our primary endpoint. So again, everyone's super happy. We give the results to the FDA, the slide deck, etc., and request for RTOR. And guess what happens 1 to 2 weeks after that request? They say no. So this was unfortunate, somewhat heartbreaking as well, because we'd put a lot of effort into this, so it was hard to take. But like I said earlier, the FDA are looking for straightforward trials and submissions in RTOR. And without going into too many specific details, what their key request was seeing longer-term safety data. We felt, the filing team had felt we'd acknowledged this in our initial submission to them. However, they wanted to see longer-term safety data, and as a result, it wasn't fit for RTOR. So this was always discussed ahead of time. We always thought that there is a potential that we won't, if they do say no, then we carry on towards the original filing timelines, and we just try and push forward with a little bit less pressure. So the filing team had submitted our responses back to their questions, and say we carried on normal, preparing for the original filing timelines. Once we got about 1.5 months or so forward, the FDA come back acknowledging our response, and guess what they said? They said they're happy with our responses and are willing to provide RTOR on the table again. So quite the emotional roller coaster working with the FDA. But it was good news. Again, like Delphine alluded to earlier, definitely not part of the plans or the timelines that we stuck to. However, yeah, we thought this was a good scenario, and we still want to take it based on how far we've progressed with the rest of the package. Of course, now the questions were being asked, because we've got a lot further, instead of just sending the top line results, could we send everything? So again, this is going back to the table and seeing, okay, what can we do in the timelines provided? And ultimately, we agreed to it, and we managed to send the pivotal study, all data for the supportive study, a small pooling, readable code, reviewer's guide, everything possible to them earlier, and we submitted it. That was great, and we also managed to get approval afterwards, and I'll show the timeline slide afterwards. So that was great news for us as well. I'll just show the timelines a little bit, just a few more seconds here, just to show you the kind of way that this went. But ultimately, you can see here where we had a period of, we had the top line results, decline of RTOR reconsidered, and this was the point here where we managed to submit all of our data to them, 6th of February. And then the full sNDA submission was on 6th of March. We also managed to submit the obligatory safety update. So we basically submitted all the data that we had available, and then that was followed by approval on the 15th of May. So again, looking from the full sNDA submission all the way to approval, it was 10 weeks. So yeah, that was our story. And over to Delphine.
So as you just learned, that we did, so both Roche and Novartis participated to RTOR pilot, but our experiences were quite different. So we tried to summarize that in this table. So just fiscally, so we had 2 pivotal studies as part of the sNDA. They were both studies compliant, so we didn't have to worry about the conversion part. At the planning phase, did we consider participating to RTOR? It was not a point for discussion, because we didn't even know it existed, and the request came from the from the FDA. Dataset-wise, so the TAU package consisted in OLS DTM and key ADAM dataset without the Delphine XML. We also benefit from regular interaction with the FDA. So we had the TC, I think, about every 2 weeks, where we can address FDA's question, and we can also share our thoughts or our interrogation. And during the RTOR process, programming team had to support a few information requests. In most of them were consisted in requests from the FDA for programming scripts, in order so that they can understand how we did produce the results and how the variables were derived. So that's probably due to the lack of defined XML. Hand over to you, Hiren.
Yeah, thanks. And on to Venclexta. So we had one pivotal study. The study submission, like I said previously, it was planned to be non-CDIS, but we went via this CDIS acceptable route. With RTOR, the biggest difference between us was we planned this at risk and front-loaded a lot of the activities. Again, another one was because of the timelines that we had in place, we managed to submit everything, every data you can imagine. So we submitted the full package. We did not have any regular TCs with the FDA. They didn't request this. And we did have information requests during RTOR, but I'll come on to that on another slide. Over to challenges.
So from Kisqali, one main challenge was the lack of RTOR knowledge. Obviously, it was brand new for every team member, so we had lots of discussion internally to know how to address this FDA's request to get things earlier. Yeah, lots of brainstorming and lots of interesting discussion, but quite challenging. More specific to programming, I think I was a bit concerned with having to work in parallel on information requests, whereas the documentation, the defined XML were not ready, because the key programmers were working had to support both high priority activities. So at some point, as you know, we don't know when information request comes, so it was had quite stress. But thanks to the hard work and the commitment of all team members, we managed to do both at the same time and don't delay the filing.
So yeah, thanks. For Venclexta, like I mentioned earlier, the biggest risk for us was actually making that decision early on to say, shall we go ahead with preparing for RTOR, even though we won't find out 4 to 5 months down the line. Ultimately, it was a smart risk, but again, it was a big discussion point early on, and there were various pros and cons to this. But yeah, that was one of the big challenges early on. Another one was learning more about CDIS. A lot of the team that we had, didn't have much CDIS experience, so it was learning whilst we were programming. So whilst we were carrying on with programming deliverables, we had to start, understanding about ADAM, about conformance checking, and doing that in parallel. So that was another challenge that we had to do in-house. And then common challenges between us were, I think Delphine alluded to this earlier with what she mentioned there, but working on multiple priorities. Again, on the project, this was the biggest thing that we were looking at, but it wasn't the only thing. So we had to look at other studies on the project, publication work, etc., and having to balance multiple priorities, which I'm sure many people have had challenges with. And another common one for people, I'm sure, that they've seen is late challenges to analyses. This was, for Venclexta and both Kisqali, this was no different. And we also had to deal with this as well. So making changes to analysis whilst we were trying to front load or carry on with RTOR submission as well. Over to opportunities, Delphine.
Sure. So from Kisqali, so we In terms of strategy, so the structure of the data sets for the supplemental NDA pivotal study followed the same, was the same as the original NDA dataset structure. So this, despite the fact that some of the standard evolved, decision was made to make it consistent. And thanks to AirTar, you have an early engagement with the FDA, and so you also have we also had the early confirmation that this was the right approach. And I think in the context of AirTar, it might have helped that the FDA was familiar with the dataset structure.
Yeah, and then on to Venclexta. So I mentioned earlier about the information requests. So on Venclexta, we only had one information request directly related to statistical programming, our team, and that was asking for further program code on a different endpoint. So we literally, lack of any information requests, and there's a variety of reasons for that, and this discussion isn't to talk about those, but one of them that we did feel could have had an impact is what we submitted and how, and that was the structure of what we submitted. So going down the SDTM route and see this acceptable route, but also the content, because of the timelines we had, we were able to submit everything. So there wasn't anything that they would have requested for in terms of data. So yeah, that was something we thought that might have helped this as well.
So when we were preparing this presentation, we also realized how AirTar was a great opportunity for programming to educate our stakeholders about the work. Obviously, other functions are aware we produce outputs, and they are very keen on getting them as early as possible, but everything that is required for outputs productions I may not be aware of. So since AirTar also focused on dataset availability, it was people were more interested in the behind-the-scene part, all the documentation, the CRT, Delphine, reviewer guide. So that was great to share our expertise with all the functions. And then another point is quite exciting to be part of a pilot, which ultimate goal is to make medication available earlier to the patients. So especially for both of us, the outcome was great. So I think it was even more rewarding for us and for the team. And also good to share our knowledge and our experience with other people internally, of course, but also externally via events such as PHUSE.
Cool. Okay, so you'll be pleased to hear this is the last slide. A lot of these recommendations are interlinked, but yeah, what we're recommending is be eSub ready at database lock. I mentioned earlier, long gone are the days where you can do all your analysis, you unblind, and then you think about eSub as an afterthought. You really need to consider doing this in parallel to your programming, and consider this as a good deadline to be ready at, especially with speed and being the key thing at this point. Explain to stakeholders what is involved. A similar point seems to what Delphine was saying. That is to make them understand how much is involved, and get rid of the myth that some people think that programmers just press a button and all the outputs appear. There's a lot more to it than that, and sometimes the education is what's needed, so that they understand that any impact or any changes can have a knock-on impact to what we do. And another recommendation is basically freezing your planned analysis, so your specifications, your list of outputs, etc. A month before database lock. Ideally even earlier than that, but the idea of this is, to try and avoid those late changes to requests. Of course, I'm saying this, and it's easier to say than to do sometimes, but putting this out there to stakeholders and to teams will make them really think about the impact that changes will have on us. So, yeah, please do consider that as well. And then Delphine?
Yeah, and one key recommendation, so again, nothing new, plan the resources ahead of time. So this is required for any kind of project, but for Airtor, this is very critical because once it's started, it goes very fast. So I would really, and we would really encourage you to use Airtor as a rationale to get the resource at the right time for your project before those Airtor process starts. And final recommendation is to, a few months before the submission, or as team is considering Airtor, to review all project deliverables, whether it's related to submission or not, and agree on a plan B if you cannot, if you have to deal with multiple priorities. What in all the project deliverables, what could be postponed or what could be even cancelled, so the submission is not jeopardized. So this discussion must happen early on, so everyone is aligned and committed to the submission success.
Finally, some references to useful links. We've also presented this at the last PHUSE conference, so there's a paper on this as well. And yeah, thank you for listening.
Thank you very much.
Thank you. We just move on to Patty now.
Okay. Lauren, can you see my screen and hear me?
Yeah. Yep, we can.
Great. Let me just put it on full. Okay. Whoops. Okay. Great. So let's just jump right in because I have a lot of material to cover in 10 minutes or so. So in October of 2019, the FDA published the revised study data technical rejection criteria, or TRC, as well as the study data technical conformance guide, the TCG, both of which introduce the simplified ts.xpt. So for the next 10 minutes or so, I am going to talk to you about the study data TRC and the simplified ts.xpt as they relate to non-clinical submissions. Although I do want to point out that both the TRC and the simplified ts.xpt do apply to both clinical and non-clinical submissions. So this is my disclaimer slide. So I'll start off by going over the technical rejection criteria, what it is and why we need it, some non-clinical considerations, and the non-clinical use of the ts.xpt file. So what is the technical rejection criteria? Well, it is an automated validation process that will be added to the existing ECT validation criteria and will enforce compliance with the study data standards requirements. And for non-clinical submissions, when activated, the TRC will currently apply only to the ECTD modules for single repeat dose and carcinogenicity studies. In the future, it will also apply to the module 4213 for safety pharmacology studies, but not currently. And I want to clarify that SEND datasets are required for studies that are modeled in an FDA-supported SEND IG and that are initiated after the SEND requirement date, even if the TRC does not currently apply to the ECTD module to which the study is being submitted. So, for example, a standalone cardiovascular safety pharmacology study that was started on April 2nd, 2020, and that was submitted to module 4213 would still require SEND datasets. So before we start discussing the TRC and the simplified ts.xpt, we first need to consider all of the different submissions within the ECTD module for modules for single repeat dose and carcinogenicity, some of which require SEND and others that do not. So we need to be able to differentiate submissions for which send data sets are not needed to make sure that they successfully pass through the TRC validation process. And for study types that are modeled in an FDA-supported SendIG, we need to be able to determine which require send and which do not based on the study start date. And to accomplish this, the TRC will look at information provided in the study tagging file as well as the ts.xpt. So the study tagging file is an ICH-defined file that accompanies all submissions in the eCTD module 4.2. And although there is a lot of information within the study tagging file, the TRC is looking for two specific things. First, it is looking for the study ID, which is used to link the study tagging file to the ts.xpt, and is also looking for the file tag, which specifically identifies the content of the document being submitted. So of the 48 ICH file tags, defined file tags, the TRC is looking for three specific file tag names, and those are preclinical study report body, or a legacy clinical study report. And these capture the study types requiring send, and they will trigger the technical rejection criteria to look for a ts.xpt file, which contains the study start date. Because of all the information that's contained in the study tagging file, there is no study start date, and the FDA needs this in a machine-readable format to determine whether or not a study requires send. So let's just take a moment and talk about the ts.xpt, which there's a full and a simplified. So when you're submitting a study that requires send, the submission will include a full ts.xpt, which is part of your send dataset. And like the simplified ts.xpt, the full ts.xpt includes the study ID and the study start date. Now, as I mentioned, there are also submissions within these three modules for which send data sets are not required. And in these cases, you would need to submit a simplified ts.xpt if you have used one of these three file tags in your study tagging file. Okay, so now let's talk a little bit more about the simplified ts.xpt, for which there are two general uses. The first is for studies that are modeled in an FDA-supported SendIG with a valid study start date that is prior to the send requirement date. And this is what your simplified ts.xpt would look like. You'd include the study ID that is in your study tagging file. You'd use the TS PARM code for study start date, and you'd include the study start date in the TS-val field in the ISO 8601 format, leaving TS-val and F blank. So there are also submissions when a study initiation date is not relevant. For example, a carcinogenicity risk assessment, which is primarily a text-based document and doesn't have any tabulated data or line listings. So this is what your simplified ts.xpt would look like, very similar to the other, except that your TS-val field is blank, and in TS-val and F, you would include NA. So Section 8 of the Study Data Technical Conformance Guide provides more information on how and when to use a simplified ts.xpt, including examples of submissions for which a simplified ts.xpt could be used. And you'll also find a link to the FDA guide for easily creating a simplified ts.xpt. So now I'm going to quickly walk through several examples of submissions for which in the eCTD modules that will hopefully give you a better understanding of how the tagging file and the ts.xpt factor into the validation process. So our first example is a 28-day study in rats, and the file tag used was preclinical study report. This study was started on November 18th, 2018, and thus required send data sets and included a full ts.xpt. So provided that it met the other four high-level validation checks, the study passed the validation process. Our next example is a 26-week study in monkeys. And in this case, the file tag used was study report body. This study was started on October 1st, 2016, and thus did not require send datasets. However, because the file tag used was study report body, the sponsor needed to include a simplified ts.xpt, which we can see over here, and the study successfully passed through the validation process. So our next two examples are studies or submissions for which send would not be required. The first is a protocol for a 26-week carcinogenicity study in transgenic mice. Here, the file tag used was protocol amendment, which accurately describes the submission. There's no relevant study start date, no send datasets required, and no ts.xpt needed because it was not one of the 3 file tags that the technical rejection criteria are looking for, and thus the submission successfully passed through the validation process. Our last example is a veterinary cardiologist study report, which is a text-based document with no line listings. In this case, the file tag used was preclinical study report. Again, no relevant study start date, no send datasets. However, because the file tag used was preclinical study report, the sponsor included a simplified ts.xpt, we can see over here, including NA in the TS val and F field, and the submission successfully passed through the validation process. So today I provided you with a very high-level overview of the TRC and the use of the simplified ts.xpt for non-clinical submissions. In November, the FDA provided an hour-long webinar on the TRC and the simplified ts.xpt that can be found on the FDA SBIA webinars website. And this webinar provides much more detailed information on the subject, as well as a live demo of the technical rejection criteria self-check worksheet, which will help you determine whether or not your submission needs a simplified ts.xpt file and contains the correct information. So if you haven't had a chance to watch it, I encourage you to do so. So in summary, the technical rejection criteria, when activated, will ensure compliance with the send requirement. The simplified ts.xpt is needed to provide the necessary machine-readable information for when send data sets are not required for a submission, and you are encouraged to look at the study data technical conformance guide for additional information on how to use and create a simplified ts.xpt. I've included some resources here. I've included the links to the study data technical conformance guide as well as the TRC. And as always, you're welcome to reach out to CDER eData with any questions. And just as a reminder, the FDA will provide 90 days notice on the ECTD webpage before activating the technical rejection criteria. So keep an eye on the ECTD webpage. And with that, I would like to thank everybody for their time and attention. And I will turn it back over to Lauren.
Brilliant. Thank you, Patty. So, Patty, we have one question for you, just whilst we've got a minute or so left. Somebody has asked, when is the technical rejection criteria being turned on?
I do not have a specific date. My recommendation is just to keep an eye on the ECTD webpage, where we'll provide 90 days notice before activating the TRC.
Great. Thank you. If anybody else has any questions, please do drop them into the chat. And whilst I wait for those, we have also a question for Hiren and Delphine. If you are submitting a repeat dosed study to the Carcinogenicity Assessment Committee, can you submit just the simplified TS, or should we submit the full send data package?
Hi, Lauren. I think that probably was a question for me. Can you repeat that again? I'm sorry.
Yep, sure. So if you are submitting a repeat dose study to the Carcinogenicity Assessment Committee, can you submit just the Simplified TS, or should we submit the full SEND data package?
So it depends on when the repeat dose study was initiated and whether or not would require SEND. If it was initiated after the SEND requirement date, you would need to submit a full SEND data set with it. If it was initiated prior to the SEND requirement date for repeat dose tox studies, then you would submit a simplified ts.xpt. I hope I answered the question. If not, please feel free to submit your questions to CDER eData.
That's great. Thank you. Okay, we haven't got any more time for further questions, but do feel free to follow up, as Patty did just say. Our next webinar will be on the 26th of February, and we'll look forward to speaking with you all then. Thank you.
Thank you, Lauren, everyone.
Thank you.
Bye-bye.